Sex determination discovered by Hanking. So there is a male heterogamete and female heterogamete. XXXY. In this, male shows heterogamy. S- For example, in human and Drosophila, XS and X0. XX represent female and X0 represent male. For example, grasshopper. It shows male heterogamete. Female heterogamete. ZZ and ZW. Here, ZZ represent homozygous male and ZW represent heterozygous female. For example, in birds, female heterogamete is seen in birds. Now, let us see sex determination in honeybee, that is haplodiploid honeybee. In honeybee, female is diploid and the male is haploid, which is also known as drone. Female has 32 chromosomes and male has 16 chromosomes because it is haploid. So, if female will undergo meiosis, it will form N, and it will undergo meiosis and form N. If they undergo mating, it forms diploid 2N, that is female. And if it does not go undergo mating, it will form N, that is, it is formed by parthenogenesis, that is, without fertilization. Now, let us see the most important topic of this chapter, that is mutation. What is mutation? Loss, deletion, or gain, insertion or duplication of a segment of a DNA is called mutation. There are two types of mutation, that is point mutation and frame shift mutation. In, for example, sickle cell anemia for point mutation. In sickle cell anemia, there is a change in single base pair, that is glutamic acid is replaced by valine. And in shift mutation, deletion or insertion of base pair of DNA caused by mutagens. For example, urease. Mutagens are the agents which cause mutation. Let us see pedigree analysis. Analysis of inheritance of trait in several generations of a family. Now there are some symbols which represents a character. That is, a square box is for male, circle is for female, diamond shape is for transgender, that is unspecified. If the box or circle is shaded, then it is affected. And if male and female is connected by bond, it is called mating. And if male and female is connected by double bond, then it is consanguine mating, that is mating between brother and sister. We can see this type of mating is seen in hemophilia. And if diamond shape and written 5, then it is 5 unaffected offspring. Now let us start with the genetic disorders. Genetic disorders are of two types, Mendelian and chromosomal. So what is the difference between Mendelian and chromosomal disorder? In Mendelian disorder, there is a change in a single base pair, or we can say in a single gene. But in chromosomal disorder, there is a change in chromosome. Either there will be a single chromosome or there will be a trisomy, monosomy, or polysomy, etc. things. Mendelian disorders are of two types, autosomal or X-linked disease. Autosomal is further divided into dominant autosomal and autosomal recessive. For example, myotonic dystrophy for the dominant autosomal. For autosomal recessive, there is sickle cell anemia, cystic fibrosis, phenylketonuria, and thalassemia. In X-linked disease, for example, color blindness and hemophilia, which is also known as royal disease. In chromosomal disorder, for example, Turner syndrome, Down syndrome, Klinefelter syndrome. Let us see each some of the disease in detail. Color blindness. In color blindness, there is a defect in red or green cones of eyes. It is sex-linked recessive disease. It occurs 8%. It occurs in 8% of males and 0.4% of females. So, it is represented by the allele X, C, and X. If we will see, mother is suffering from color blindness and father is not suffering from. So carrier may be daughter or carrier may be son. Hemophilia. Hemophilia is a sex-linked recessive disease. It is also known as royal disease because Queen Victoria was suffering from this disease. Female become hemophilic is extremely rare and hemophilic males are unviable at later stage. In hemophilia, there is a lack of factor 8, factor 9 and factor 11. So, if there is a lack of factor 8, that is anti-hemophilic factor, then it will cause hemophilia. Sickle cell anemia, qualitative disorder. It is autosomal recessive disorder, substitution of glutamic acid by valine at 6th position of beta globulin protein of HB. GAG, that is glutamic acid, is replaced by valine, that is GOG, by mRNA. Mutant HB undergo polymerization under low O2 tension. Shape of RBC changes to elongated sickle-like. Sickle cell anemia occurs in 11th pair of chromosome. Now let us see thalassemia. Thalassemia is a quantitative disorder. So there are two types of thalassemia, that is alpha thalassemia and beta thalassemia. Let us see alpha thalassemia. Reduced synthesis of alpha globulin chain. That is, if we will see hemoglobin is made up of 2 alpha and 2 beta globulin chains. So in alpha thalassemia, it reduces the synthesis of alpha globulin chain and closely linked HbA1 and HbA2 genes of chromosome 16 are affected. This is necessary to remember that it happens in chromosome 16. Now, beta thalassemia. It reduces the synthesis of beta globulin chain and HBB gene of chromosome 11th is affected. Now, why it is known as quantitative disorder? Because the globulin protein of hemoglobin is reduced. Now we will see what is aneuploidy and polyploidy. If we will see aneuploidy, A means decrease and poly means increase. So gain or loss of a chromosome is known as aneuploidy. Increase in a whole set of chromosomes is known as polyploidy. Let us see chromosomal disorders. Down syndrome. In Down syndrome, additional copy of a chromosome 21 is seen, that is trisomy on 21st pair of chromosome. It was described by Langdon Down in 1866, and we see effects like short stature, forward torque, palm crease, and mental retardation is seen. Klinefelter syndrome. It is seen in 23rd pair, that is trisomy of 23rd pair, that is there are 47 chromosomes. Overall, we can see overall masculine development. However, gynecomastia is also there. Gynecomastia means development of breast. Klinefelter syndrome is seen in males and the sexual characters are underdeveloped. Let us see Turner syndrome. Turner syndrome has also underdeveloped sexual characters. It is seen in females. Now there is 44 plus X0, that is we can say 45 chromosome is present and there is an absence of one X chromosome. Female with only X chromosome, therefore she is sterile, and rudimentary ovaries, no secondary sexual characters is seen.